in cells of the immune system, signaling leads to activation of cell type specific immune signaling. The innate immune response starts with pathogen recognition, and although the innate immunity is fast and usually takes 4 hours to be established, it is non-specific and does not have memory. But what's really important is that it identifies the self from non-self, thankfully because pathogens have fragments or regions which are not present in human cells. Now moving on, just to demonstrate how pathogen recognition goes, for example, there is a break in the vessel and the pathogen infiltrates. Our body is actually ready to face these pathogens as we have cells that circulate in the bloodstream to protect us from these pathogens, and these will include your macrophages and your natural killer cells. Aside from these cells readily circulating in the body, actively activating or attacking the pathogens, we also then release chemical factors known as your cytokines to further signal other lymphocytes to go into the area, and what they do is basically signal to each other and to other cells to come help the soldiers fighting the invaders so that we have a bigger chance of surviving. Now, as I have mentioned earlier, there are unique regions in these pathogens which are recognized by our receptors. And this would include your flagella or even your flagellin, lipopolysaccharides, which are endotoxins present in gram-negative bacteria, peptidoglycans present in gram-positive bacteria, RNA in viruses, and collectively we call this your PAMPs or your PAMPs, pathogen-associated molecular patterns which are found in pathogens. Then, of course, to recognize the PAMPs, you have PRRs or your pathogen recognition receptors, and these would include TLR or your toll-like receptors, NOD or your nucleotide binding oligomerization domain-like receptors. And what these NODs do is that they recognize peptidoglycan motifs from bacterial cell which consists of N-acetylglucosamine and N-acetylmuramic acid. Rig-I-like receptors or your retinoic inducible gene I-like receptors or RNR, as opposed to other receptors such as TLRs, which recognize extracellular viruses. RNRs serve to recognize viruses that have already entered into the cytoplasm of a cell. Particularly, they read your double-stranded RNA of the viruses. Now, going back to your toll-like receptors, toll-like receptors are usually expressed on sentinel cells such as macrophages, dendritic cells that recognize structurally conserved molecules derived from microbes. Now, toll-like receptors 1 and 2 recognize peptidoglycan and triacyl lipoproteins specific for gram-positive bacteria. 2 and 6 recognizes DALP. 4 recognizes lipopolysaccharides. 5 recognize flagella or flagellin. And 3 and 7 recognize RNA for viruses. So as mentioned, the signaling is to ask for help from the adaptive immunity. Because what happens here is that the TLR recognizes the signals to activate transcription factors to transcribe interleukins, histamines, and bradykinin and other cytokines to initiate inflammatory response. Now, your inflammatory response is actually kickstarted by innate immune cells, as mentioned, pro-inflammatory cytokines, and complement. It acts as a bridging mechanism to localize and contain the infection in the period from about 4 to 96 hours after its onset, when the innate immune system is running out of steam and the specific cellular response is trying to get going. Now, the main features of this process are the innate immune system and inflammatory response can only hold off an infection for so long. Ultimately, a specific immune response needs to be activated. This is done via antigen presentation to the adaptive immune system. The immune response also depends on how the antigen has entered. If the antigen is a whole pathogen, it is engulfed by the macrophage and it becomes an antigen presenting cell which fragments this antigen and showcases antigen to let the body know that there is a pathogen that needs to be fought. This process activates the helper T cell and further activate either the B cell or cytotoxic T cell. Now, if it is a free antigen, meaning it's just a portion of the pathogen, the B cell will be directly activated and turned into a plasma cell which will then produce antibodies and also activate the complement system or be killed by any of the phagocytic cells. If the antigen is one of our cells, the normal cell modifies to the signal the cytotoxic T cell and gets activated and kills the infected cell through apoptosis. Whenever this happens, the immune system stores information about the pathogen through the memory B cell and the memory helper T cell so that after another attack from the same pathogen, there will be a faster response because it can directly be attacked by the B cells and the T cells. Now, on the left side is the humoral mediated immunity, as they are mediated by antibodies, while on the one on the right side is cell mediated response. Now, let's take a look at this bit a bit closer. So, as mentioned, your antigens act as ligands for BCR, and what happens is that this is going to be phagocytized by the B cell, and the antigens will become degraded in the lysosomes, and we come up with this peptide, which will bind with the MHC and become ejected outside the B cell, then consequently interact with the T cell. And this cell-to-cell interaction between the B cell and the T cell will result to signal transduction. Now, after stimulation of the T cell receptor, what happens when the antigen binds to the T cell receptor is that a phosphorylation cascade follows. As we know, in a phosphorylation cascade, it is an interaction of string of kinases activating one another in a chain reaction. Once the IP3, a ligand which will bind to the ER cell membrane, which is an ion-gated channel, and since this is an ion-gated channel, once opened up, will allow for the release of calcium into the cytoplasm. And what will happen is calcium will participate in a series of reactions activating the NFAT, which is a nuclear transcription factors, and initiate transcription to allow T cell to differentiate into a helper T cell. Now, in summary, immune response starts with pathogen recognition by cells of the innate immune system with cytokine release, complement activation, and phagocytosis of the antigens. The innate immune system then triggers an acute inflammatory response to contain the infection, and meanwhile, antigen presentation takes place with the activation of specific T helper cells. Your CD4 helper T cells then coordinate a targeted antigen-specific immune response involving two adaptive cell systems, humoral immunity from B cells and antibodies, and cell-mediated immunity from cytotoxic CD8.